like a love-hate relationship with introducing my students to the exit seminar. I think the reason is that it seems like no matter what you say, it's always either too much or simultaneously somehow not enough. And it's either it's always like over prepared, but at the same time like just not prepared enough. Like it always catches me by surprise. It's always like either too serious or not serious enough, and like simultaneously all those things at the same time. So it is what it is. I'll do my best. So I'm here to, it's my pleasure to introduce Tracie for her exit seminar. The details, Tracie grew up in North Carolina, and she did her undergraduate degree at UNC Chapel Hill. She worked on the phenology of arthropods and migrating birds with Alan Hurlburt as an undergrad, and kind of remarkably at the same time, she completed a degree in studio art. She not only completed a degree in both biology and studio art, she completed 2 honors theses in studio art and biology. After she graduated, Tracie continued to pursue both art and science. She did field work in Minnesota on the remarkably long-running Echinacea project, and somehow found the time to develop multiple art exhibitions in Chicago and in North Carolina. When Tracie arrived at UC Davis, she was full of curiosity and questions. I actually went back to your application and read some of what you wrote. You wrote, "I hope to be a creative graduate student that is not afraid to make mistakes and learn from them, since this will make me a stronger scientist in the long run." However, it was very quickly clear to me that Tracie was never going to be one to study butterflies and wildflowers. Given the range of her own research, she was very immediately attracted to the dark, often overlooked undertakers of the undergrowth, the burying beetles. These are the scavengers that regulate the transition between life and death, and then back again. And their ecology is fascinating. Tracie's done a remarkable dissertation elucidating the previously unknown role of coastal fog in the ecology of these burying beetles, and I'll let her tell that story. But I will note that there to watch her develop as a person and as a scientist, and eventually to outgrow the limits of our graduate program. So I know she will go on to do great things, and with no further ado, please welcome Tracie Davis.
Well, thank you, everyone. Thank you, Louis, for that introduction. Yeah, did not know I'd be getting quotes from my 6-year-old application, almost 7-year-old application. But yes, thanks for everything. Yeah, so title of my exit seminar today is Beetles and Buckets. You'll note that that is a theme throughout my experiments. Testing the effects of resource ephemerality on an obligate scavenger. And I do want to start with some land acknowledgements. So the first is just the UC Davis land acknowledgement, which was written and approved by the Patwin people. And it goes, "We should take a moment to acknowledge the land on which we are gathered. For thousands of years, this land has been the home of Patwin people. Today, there are 3 federally recognized Patwin tribes: the Cotchal-Dihi Band of Wintun Indians of the Calusa Indian Community, the Quetzal-Dihi Wintun Nation, and the Yocha-Dihi Wintun Nation. And the Patwin people have remained committed to the stewardship of this land over many centuries. It has been cherished and protected as elders have instructed the young through generations. So we are honored and grateful to be here today on their traditional lands. Additionally, my research that I will be talking about today was held at the Bodega Marine Reserve in what is now known as Bodega Bay, California. And this peninsula along the California coast is the traditional land of the Coast Miwok, and specifically the Bodega Miwok or Olomenko. So before colonization, the Olomenko lived on this peninsula for thousands of years, and then after genocide, all but eradicated the Olomenko. In the early 1800s, settlers have used that land for farming, grazing, and pasture. And the University of California that acquired that property specifically in 1963. So that land was cherished and protected long before settlers arrived. And then finally, I do just want to make a note. So additionally, like as an ecologist, I do believe that it's important to not ignore the historical and ongoing removal of Native people from their lands. This is a process that includes both genocide and ecocide. This continues today in places such as Palestine and is perpetrated by empires and their westernized institutions like universities that uphold this settler colonial order globally. UC Davis continues to facilitate land theft, and both in its research partnerships, its contracts with Israeli institutions, military forces. And so because both higher education and research often play a major role and are complicit in genocide and ecocide, I just strongly believe that as ecologists and scientists generally, we should do more to acknowledge and act against the colonial nature of our own work. So part of what we can do as scientists is to use our creativity to reimagine what it means for us to embrace Indigenous sovereignty and land back, both here, Turtle Island, and in Palestine. So thank you for listening. Okay, so getting into my kind of general intro. So ecologists have known for a while that resources are structured both in space and in time. So in this 2D representation of space, you might imagine resources only available in certain patches. And then when we think about resource availability in time, rather than being constant, a resource often has different levels of availability over a period of time. And so, for example, let's say you are this beautiful yellow-faced bumblebee and you live in this coastal prairie of California. You enjoy collecting pollen from lupins and pollen and nectar from lupins and from seaside daisies. And these flowering resources are only available in patches in space, but they're also not available constantly in time. So those same patches are ephemeral in time as the pollen and nectar becomes available and then is removed or consumed. So there has been less of a focus on the time component throughout ecology, but a few key frameworks do stand out. First are resource pulses. These are rare short-lived events that are large in magnitude and have direct and or indirect effects on both the numerical and the aggregative responses of consumers. And the key component here is their brevity and magnitude in time. And then one well-known example of this, Louis' dissertation research, are resource pulses of the emergence. Resource pulses is the emergence of periodical cicadas. So they're this kind of ephemeral superabundance, and this has consequences for both below ground and above ground interactions. And then there's also this ephemeral resource patch framework, which these are essentially just repeated smaller magnitude resource pulses in explicit space. So usually there are specific community assemblages that use them, and they are often used up within one generation time of the most common consumers. So the authors of this review paper, Butterworth et al., argued that ephemeral resource patches are hugely important for shaping species adaptations and community diversity. And examples of these are fruit flies on bird dung, frogs mating in an ephemeral stream, and also ants on a honeybee carcass. So thinking at the scale of the individual patch, what influences the ephemerality of a resource in time? And so one obvious way that a resource might stop being available is if it gets used up by another consumer first. So going back to our bumblebee example, we might imagine that this lupin's pollen was already collected by another bumblebee. So this graph then kind of shifts depending on whether or not there is competition. Another way we might expect resource availability in time to shrink is due to shifts in environmental variables, like temperature and moisture. And these warmer and drier conditions might shrink this window of resource availability in time. An example of this could be nectaries in the seaside daisy actually drying out faster when it's warmer and drier. So less energy than available for those bumblebees. And shifts in resource ephemerality like this are especially likely under climate change. Here are just a few quick examples. Spittlebugs needing moist refugia, climate change induced phenological mismatch between birds and the food they feed their nestlings. Also salmon not having enough cool water to migrate through because of decreases in fall. So how does this increased ephemerality affect the consumer? And I got, let's see how far, I got in like almost 10 minutes in without mentioning anything about beetles. So here we go. So yeah, happens to be that burying beetles are specialists of ephemeral resources. They're in genus Nigrophorus. So they locate a small carcass that they then use for reproduction. Cookies back there look very similar to this mouse here. And so a pair will work together to bury the carcass into the soil to protect it from competitors. The female will then lay eggs, and both parents will participate in parental care by regurgitating pieces of the carcass to feed the growing larvae. So specializing in this way makes them obligate scavengers. And I will show like a minute-ish video clip of this film that was made by Deep Look with beetles that were used in my own experiments. So I was very proud of their acting abilities. But yeah, and I guess brief content warning, there will be a carcass in this video. Hopefully it's cool with people, but if you need to close your eyes. Okay, so I think the sound itself, like of the person talking, isn't playing, which is cool. So basically she's just showing, like explaining this process where this is a single beetle doing all of this hauling. They highlighted their interesting antenna at the beginning, which they use to like find the carcass via scent. Yeah, so this hauling is happening. And then there's really silly music where things kind of get like jazzy in a moment, like it's coming up soon. Here it is. Jazzy music, something else is going on. So they're like, this is happening. So yeah, hooking up with a mate, and they're like, oh, this is one way to bond on a date. They say that. Like, this is like silly things that happen. So they roll the ball into a carcass. This is because, you know, lower the surface area, less microbes, like keep it preserved. Takes on this kind of form here. And oh yeah, there's a really fun part where they show like their anal secretions, which are also antimicrobial. That's coming up soon. Well, they'll like highlight it with a little circle. Here it comes. Oh yes. So, and then beautiful larvae are hatching at their feet from the carcass. Those are older larvae there. Yes, I hope you enjoyed that. Yeah. So these are kind of the main players in the system that I work in. Today I will only be focusing on a few of these. I'll briefly go over. So the first is Nigrophorus scutula. This is the yellow belly bearing beetle. It will be my focal species throughout my talk. They use these small carcasses like the mouse shown here for their reproduction. The next is this genus Heterosilpha. This is a more generalist carrion beetle. They also will feed from small carcasses and will lay their eggs nearby, so their larvae can feed, but they don't specialize as much. They can feed and reproduce with a wider range of resources, so are less specialist. And so in my first chapter, I will be including them as competitors since they actually limit this resource's availability. And I use experimental chambers in the field, basically just buckets partially submerged below ground. I add beetle pairs and then carcass treatments and then close it up with a mesh lid. The chambers are secured with a cinder block so that vertebrate scavengers cannot rip the lid off and steal the mouse carcass. And then I return later to check for burial, for reproduction, for survival of those original. block cake in the back, I do use a lot of cinder blocks. Yeah, inspired by Maddie's seminar last week with like little photos of kind of the process. Here are just some of my key tools. Of course, a post hole digger, lots of hole digging, flag pins, buckets with modified lids, cinder block, Icelandic grocery store bonus bag. This is like really good for holding lots of buckets. And it's like been patched up with duct tape over the years and is now going into retirement after many field seasons. So yeah, I did all my chapters here at the Bodega Marine Reserve. This is on Coast Miwok land along this California coast here. And these beetles are pretty common here in the coastal prairie. And so at this site, some days are sunny and warm, and some days are foggy and cool. And sometimes both of these conditions happen within one single day or even multiple times over the course of a day. And so with climate change, we might actually expect more heat waves, less fog in environments like this. And these environmental conditions can affect that resource availability in time as well. So before I go into each of my 3 chapters, I do want to briefly introduce this art exhibition that I was able to organize through a Belinsky Fellowship. And I will show a few of the pieces kind of as I go through my experiments as they're relevant. So briefly, yeah, I collaborated with these 9 artists here to do an exhibition out in the field at Bodega. This was in June 2023. And yeah, basically spent that year leading up to it, spending a lot of time with each of the artists involved. So we met frequently to talk about ideas. We did field work together. We read papers about scavenging and generally just kind of learned and thought together. And I mentioned earlier how I've been doing my experiments with these buckets that are submerged in the soil. So I've been calling these experimental chambers microcosms. This is after this paper in 2016 by Paul E. Hopwood et al., where they call their experimental chambers microcosms as like a play off of mesocosm or microcosm, with that first component, micro, with an N, coming from the bearing beetle genus name, which is Necrophorus. And also the root cosm can mean world, can mean order, universe. So in this way, we can kind of think of each microcosm as encapsulating an entire universe, answer to an ecological question, the life's work of this unseen beetle, birth and death, changing climate, the passing of time, etc. So this idea of microcosm was kind of just like the main prompt I was giving these artists as they developed their loose interpretation of a microcosm. So like some kind of portal into the overlooked world of scavengers and below ground processes. And so the setting was out here in the coastal prairie. And visitors were given this map so they could find each of the pieces. And yeah, I will take you through a few of those. I'm going to start quickly with one of those pieces. This is Kelly O'Leary. Kelly's piece involved multiple ceramic structures created from clay collected at a nearby beach. She included cinder blocks and flag pins in her installation. Cinder blocks are so inspiring to people. Yeah, 2 key tools that I use. So cinder blocks to protect my scavengers from pilfers. Find these again. And these little assemblages that she made were kind of scattered over the exhibition area. You might even say kind of randomized. And each clay piece involved like a hole or a portal of some kind. And I want to read what Kelly wrote about her work. The studies conducted at this reserve have left traces upon the landscape that collectively form a sort of archaeology of ecological research. The physical remnants of the inquiries made are a constellation of holes, buried PVC pipe, flags, wire, cinder blocks, buckets, cement, and rope. Using clay that was foraged, these earthen chambers are inspired by the hollows that the bearing beetle carve and also allude to the researcher's method to trap and study the species. They become speculative artifacts of a subterranean world as experienced by both the researchers and the researched. So with that as an intro to my field process, I will take you through some of what I have found over the years. So a reminder that my big question is about how resource ephemerality affects the consumer, in this case, bearing beetles. And for each of my chapters, my response variables were reproductive output, burial, and survival. And my first chapter involves carcass dryness and competition as my manipulated variables. The second considers the buffering effects of leaf litter. And then the third takes a closer look at the effects of shifts in fog availability. So we'll start with chapter 1. This was an experiment on the effects of carcass dryness and competition, how that affected reproduction and burial. So my big question here is, what is the relative importance of environmental conditions and competition on resource availability for a consumer? And of course, this is likely to depend on the strength of each of these factors, but under realistic conditions that a consumer might experience, which of these plays a greater role? And so as mentioned in my intro, a main competitor of Necrophorus is Heterosulfa. And this is a generous carrying beetle that feeds on many types of detritus, and it has the capacity to. And my environmental condition is moisture, as I was thinking about how much variation there is in fog on the reserve, and how this might also affect resource availability and time. So to actually make my manipulations in moisture, I dehydrated mouse carcasses in a food dehydrator. But how representative is this of natural drying out at this site? This was Gary's idea, by the way, the food dehydrator, way back in the day. It's like, oh, perfect, delicious. So yeah, but actually, how representative is this of actually drying out at this site? And so I did a kind of proof of concept study to measure carcass dehydration in the field by just setting out dead mice and returning multiple times to weigh them. So along this x-axis here, we just have time, and then the y is like percent carcass mass remaining. So all mice will start at 100% and just decrease with time. And so this is what mass loss looks like over about 4 days. So we see like pretty significant loss. And the red line here shows approximately when the carcasses lost 25% of their mass. And this was the average percent loss that I used for my treatment carcasses in my experiment. So mimicking 1 to 2 days of naturally drying out. Project, these fog collectors were collecting fog measurements. So I could see if more mass was lost when there was less fog. And the x-axis has the average fog based on those collectors in milliliters per hour, and then the rate of carcass mass change in grams per hour. And so on the left half here, when there is less fog, there's a greater rate of mass loss, so it's more negative. And on these super foggy nights up on the right half, there's actually a little bit of mass gained for those carcasses. So I collected Necrophorus in pitfall traps. I paired them up, placed them into 63 chambers and transects across the reserve. So a quarter received a semi-fresh mouse with no competition. Another quarter received a 25% dehydrated mouse. And then another quarter received competition in addition to a fresher mouse or control mouse. And then another quarter got the worst deal, got a dried out mouse with competition as well. And this competition here is realistic based on like pitfall trapping at the site. So I would usually find on average like 2 Heterosulfa per, like if there was a Necrophorus present. And I want to give a huge thank you to my amazing intern. This was in 2022, Leo. We together, well, he carried more cinder blocks than I did, but we carried lots of these 39-pound cinder blocks up the hills of the South Bluffs. We would pause to take breaks together, and we would sit on our cinder blocks, and Leo would tell me about the dun beetles of France. So it was actually it was a really great summer. And yeah. I also want to thank Gwen. She was another amazing undergrad who helped in the field multiple times that summer. Give the big reveal. This plot will show the mean number of offspring in each of these treatment combinations. And we actually see a huge effect of carcass dryness. So almost basically only 1 pair reproduced if they had this dried out carcass. And for those pairs that did reproduce, competition did negatively affect the number of offspring. And this will be a similar plot, but I will show whether the Necrophorus pair buried the carcass. So dark green is buried with successful reproduction. The lighter green is just buried with no reproduction, and then no burial in purple. And so all of the control carcasses were buried, whereas most of the partially dried ones were not. We do see a difference in burial for undried with and without competition. So potentially this is indicating that the necrophorists were more likely to bury. So overall, we find a greater effect of carcass dryness on reproduction compared to competition. So the green points here show how much each of these variables affected just the probability of reproduction at all. And then the purple points show the effect sizes of those same variables on the number of offspring, and that's just for a subset, like for the pairs that actually reproduced. So we can look at the effect sizes of these 2 points here. Carcass dryness had a large effect on the probability of reproduction, and presence of competition had a relatively small effect on number of offspring produced. So yeah, so carcass dryness inhibited both burial and reproduction outright, while competition limited the number of offspring for reproducing pairs. So this could suggest that environmental moisture could affect the availability of usable carcasses kind of over the landscape, while competition then reduces reproductive output at a smaller spatial scale once a usable carcass has been secured. So in summary, we can say that environmental conditions more strongly constrain resource availability compared to competition, in this system at least. And our results suggest that the window for successful reproduction on a carcass could be substantially shorter under increasingly dry conditions, reducing opportunities for bearing beetle reproduction. And then more broadly, our results kind of have implications for ecosystems that are getting drier with climate change, and for guilds of species like scavengers that rely on these ephemeral resources specifically. This was published earlier this year in Ecology. Okay, so next up, chapter 2. So this was an experiment also considering carcass dryness, but this time with leaf litter. Before I get into the details, I want to show you all Sophronia's piece. Sophronia created this beautiful geologic quilt on the landscape. Glaze, earthenware, glass, stones from a bunch of places where the artist worked. So Chicago, Baltimore, Steuben, Waterville, Bodega Bay. And she writes, this site-specific installation is a geologic quilt foraged from sedimentary deposits, roadsides, and riverbeds. And Sophronia's piece is relevant in that it depicts a mosaic or variation over a landscape. Similarly, there is a mosaic of resources in space in the coastal prairie of Bodega. So one resource that is quite patchy is the bush lupin, seen here in these kind of gray-green splotches you can see kind of throughout. And tussock moth caterpillars feed on the leaves of these lupins, and since they are such messy eaters, they produce a lot of leaf litter below the plants. And in fact, the larger the abundance of tussock moths on a lupin bush, the greater litter depth below it. So we have this mosaic of lupins, and then a further mosaic of tussock moth abundance affecting leaf litter depth. And in some of my early, like have a string attaching them to a flag pin, I would actually find that necrophorists would pull the carcass a few feet so that it could be buried underneath a lupin. So my question for this chapter is, can resources in space actually buffer the effects of resource scarcity in time? And in this case, does lupin leaf litter kind of provide an insulating effect that slows the drying of a carcass resource? So this scarcity in time that we might see due to drying and warming, if lupin litter is available, can this squishing of availability in time be prevented and allowing more survival reproduction of our varied beetles? And so to test this, I use the same necrocosm setup as my first chapter, but have different manipulations. So instead of a 2x2 factorial, we actually have a range of carcass dryness from 0 to 25%. So I already know from my first chapter that 25% is like the limit, like reproduction does not really happen there. So I wanted to look across this range to see where lupin leaf litter might actually have a buffering effect on quality. And Sydney was a huge help with this field experiment. She was a part of the Bodega Marine Lab, Santa Rosa Junior College internship program. Here she is in the field with a little mouse swing. This is what we use to like weigh carcasses in the field. And so for these results figures, the x-axis will be percent carcass dried from 0 to 25. And solid brown lines will show experimental chambers without any lupin leaf litter. And then dashed green lines will show those with that litter addition. And I'm starting here with survival of the males and females. So I hypothesized that basically survival would decrease as the mouse carcass gets drier, but that leaf litter would have this buffering effect. So higher survival with leaf litter. And then I predicted a potentially stronger effect for male survival, or just lower survival in these bare chambers, since it cannot hide as well from the female, who may decide that it's time to like kill him when he's no longer useful. So that's kind of my prediction there. And I found that leaf litter does indeed buffer the effects of carcass dryness on survival for both sexes. Interestingly, there is this interaction for females where they actually seem to do better with control carcasses when it's just a bare environment. But yeah, as carcasses get drier, leaf litter does become more important for that female's survival. And this is the case across all carcasses for males. And then I had similar hypotheses for burial and reproductive likelihood. So I expected decreasing rates of burial and reproductive likelihood as mouse carcasses got drier, but that both rates would be higher under leaf litter. And I also generally expected reproductive likelihood to be lower overall than burial, just since other things can go wrong. You can bury the carcass, but there may be other things that inhibit reproduction to be successful. So it's clear here that carcass dryness has a negative effect on burial and reproduction, but we learned that from my first chapter. My sample size here was too low, and I probably checked chambers too soon to say anything about the effects of litter. So for now, these results remain somewhat inconclusive. But as a small follow-up to this experiment, I did a mini experiment where I put out about 25 mouse carcasses, half with lupin litter and half with no litter cover. They were in these cages to kind of partially deter scavengers from stealing them. And then but these also had holes available so the beetles could get in. And then I checked them every day for like about a dozen days to measure weight loss and also to look for evidence of burial. So on the left here, I will show the percent mass of carcasses remaining over time. And the lines are color-coded, similarly for bare and litter. And so the effect of litter, it's subtle, it's insignificant, but there's a hint at different rates of drying that might be evident if I did a larger experiment. And on the right, I will actually show the likelihood the carcass was buried by necrophorus for each treatment, based on data where I either saw necrophorus in the process of being buried by a beetle or found a partially buried carcass. This is just what that looks like. So you can kind of see they're usually buried head first. And so we have the two legs sticking up and the little tail. But honestly, more often I actually find just the little tail sticking out, probably because it's not really a useful resource. So they're not even worried about getting that below ground. Yeah, so the figure on the right here will be probability that I found that a carcass with evidence like that. And though there's not a significant difference between these two curves, there is an interesting trend. So at first, the necrophorus seem to be finding and starting to bury those bare carcasses. But then as time goes on, there is a much greater likelihood that burial activity is found for carcasses that have leaf litter. So I'm just speculating on what this could mean, but it might be that carcasses with leaf litter covers stay higher quality for longer, effectively making that resource less ephemeral. And so for this chapter, we can say that resources in space buffer some of the effects of resource scarcity in time. Specifically, we saw this with survival. Okay, so now I want to move on to chapter 3, a fog addition experiment. And I want to show Rosemary's piece, one of my favorites. So Rosemary came out to the field site and was interested in fog collectors. that I'd helped set up. You can kind of see them. They were in line with her fog collector over here. And Rosemary was inspired by, the structure of the fog collectors, but also the mystery and the magic of a microphorus pupa. So this is like this in-between period of development. And she created this metal structure based on a microphorus pupa and then covered it in a mesh similar to that of a fog collector. So you can see fog actually like condensing on it onto the mesh here. And over time, gravity pulled this water and it collected on the mesh towards the abdomen, so that it began to drip. You can it's almost like an anal secretion. And it would drip into the trough below. And so you can kind of see the water droplet forming with the debris swirling around right before it kind of falls into that trough. So Rosemary writes, Encyclia imagosis, common name sea-dew, is a human scale fog collector that takes the form of a microphorus beetle pupa. Interested in how fog moves, how plants and animals adapt and collect occult precipitation, and how humans navigate with the distant call of a foghorn, sea-dew hones attention on seeing what the place does and how the body is embedded in that being. So Rosemary's fog collector kind of hints at the results I will find in this chapter. So to start, we know that fog has been decreasing along the Northern California coast. This figure shows fog frequency in black, and then the highly correlated contrast between inland and coastal daily maximum near surface temperatures, so indicating less and less fog over the last century. So coming back to this framework, you might think of fog as a resource that is decreasing in its availability over time. And at Bodega specifically, there's already a lot of variation in fog deposition depending on the day. This is just an example month, June in 2023. So there's days where there's a lot of fog deposited and then days where there's basically none. And so with fog generally decreasing in the area, I decided to mimic this variation in an experiment to kind of project what might happen as fog continues to decrease. And in particular, I'm considering how the shrinking of fog availability in time might affect resource scarcity and consumer responses. So the question here is, do subtle shifts in fog during the initial period of carcass discovery have a noticeable effect on burial, reproduction, and survival? And in this experiment, I had 3 different cohorts that I checked after different periods of time. So here, these drops represent the fog manipulation that I conducted on the day the necrocosm was set up, and then the 2 days after. So these fog addition chambers received squirt bottle fog on all 3 days, and controls did not. And then I checked for my response variables on the same day at the end for all cohorts. And I did this so that I could get at effects that might be more immediate. So something we might see after just 7 days versus effects that are delayed that we don't see after until most of the reproductive process is over, so after 27 days. So here's my experimental setup with the sample sizes in each cohort. And I want to just briefly talk about the realism for my fog manipulation. So these are kind of the totals deposited over the day. And basically, with calculations from the fog collectors, etc., my control manipulations represent basically 0 to low fog days. And the fog addition manipulations represent pretty low fog days as well, but just 3 consecutive days of fog. And before I show my results, I do want to thank these 2 undergrads, both Melanie and Kai, Melanie's here, for everything they've helped with in the lab over the past 1.5 years. So they processed beetle photos for this experiment, but also a bunch of other projects that I've been working on that I'm not even able to show today. And I'm just so glad I got to work with them both. Sorry. So I will start with burial and reproduction for my results. So in these figures, the x-axis is chamber age or time in days. And we can imagine these proportions changing in time based on those different cohorts. So fog addition lines will be solid and control will be dotted. So in my hypotheses, I expected pairs receiving fog addition in those first few days to perceive a higher quality environment and resource, making them more likely to bury the carcass that's available. So after just 7 days. And I expected this difference to then lessen with time as pairs without fog addition, bury their carcass because it's kind of all that's available to them in the chambers. And then for reproduction, 7 days is usually not enough time for reproduction to have happened. But as time goes on, I expected greater reproduction rate for fog addition compared to control. And so we do see this immediate effect of fog on burial. And though there looks to be a difference in proportion reproduced after 27 days, this was not significant. But what about actual number of larvae? So these results will be a bar chart with bars for each treatment and in the 2 different chamber ages, leaving out 7 days just because no reproduction happens after 7 days. So here I expected to see greater numbers of larvae for fog addition chambers, as that moisture received at the beginning of the burial process might have been sequestered away and then used when the carcass was pulled below ground and the larvae were hatching and feeding. And then additionally, I expected numbers of larvae to be higher after 27 days compared to 19, just because they've had more time. And so we do see an effect of fog treatment. Brood sizes were bigger in fog addition chambers, regardless of chamber age. However, the 27-day-old chambers do seem to be driving this result. And then I will briefly show my results for male and female survival. So I expected higher survival in fog addition chambers compared to control. And the results here are not quite as clear, but there was a significant difference in survival across treatments. So when survival was higher in fog addition chambers, that we did see marginally or significant effects of fog. So in summary, with decreasing fog in coastal areas where burying beetles are common and where they rely on summer moisture during their breeding period, we can expect negative and lasting effects on reproduction and survival. And for organisms across the globe that are sensitive to moisture availability in time, burying beetles kind of serve as a warning story for challenges related to moisture scarcity. So in conclusion, I came in interested in resource scarcity in space and time. And particularly, I was curious about what influences ephemerality, competition versus temperature and moisture. And I did 3 experiments considering how increased ephemerality affects these response variables. So chapter 1, basically I found that a partially dried carcass pretty much completely inhibits reproduction, also limits burial. In chapter 2, I found that leaf litter can buffer some of these environmental effects, especially for survival. And then in chapter 3, I showed that even a subtle manipulation in fog during these initial days of carcass discovery does have a substantial effect on reproductive output. So altogether, these beetles have taught us about resource scarcity, how a changing climate could then further exacerbate those effects. And I want to just show one last piece before finishing up. So this is Alejandra's piece. It's titled Microcosm Playground. And so Alejandra was very interested in the materiality of the place, all the plastic mesh, etc. She enjoyed looking at the necrophorus up close under a microscope, and the algae pieces kind of almost plastic-like. So she spent time studying all of these things and came up with a series of sculptures, which were created from leftover plastic mesh. And she writes, the sculptures reflect on the craft and practice of science, creating empty measurable gridded spaces where the object and subject of research can meet to amaze each other. So that kind of sums up everything, the beetles amazing me, and then hopefully in some way I'm also amazing them as well. And then I just want to say, this is a quick preview of the 5 other pieces in the exhibition. If you're interested, I have mini catalogs if you want to check these out. And this is my huge big slide, just lots of text of everyone I want to thank. I will highlight a few. First, I need to start with my family. So my parents, my mom is here, my dad's on Zoom, Thank you. I love you both. I obviously could not have done any of this without you. Same for my brother and my kitty, Minna. They, have been supporting me through my whole life. So really important to thank them. And to thank Louis, my advisor. I don't have us as well prepared. Thank you as Louis, but Louis's been there for the last 6+ years and has been such support and joy to work with. I need to thank Jackie at Bodega. She has been basically like my field mom, like every summer for 6 summers. She's been so supportive. She sings little Beatles songs with me. Like, I really love Jackie, so just needed to highlight her. And then I gotta thank all of my field help, tons of folks who aren't listed here, the Yang Lab, past and present. It's been amazing to be with you all. My dissertation committee, so that's Louis, Rick, Marcel, and Tom, all of CPB, the Population Biology grad group, my amazing cohort, the storefront office, my physical therapist, who I've been working with for almost 5 years, been very important for grad school for me. All the amazing students I got to teach while TAing, my funding sources, the friends that I made, and then I do have pictures, and I'm just gonna show them. I love you all so much. This is some of these pictures were like babies, but it's way back in the day. Field work together, exploring together, wearing costumes together. I have 2 more slides with photos of people. I like doing art together, pretending like I'm a geneticist with Maddie, over there. So wonderful. Going to the Fishitarian over here. All of my friends. I love you all so much. And then of course I have to have a whole slide for my cat. She's so cute. And also her boyfriend, which is Piggy there in the corner. They are lovers via under the door. And then of course I have to thank the Beetles, the Lubins, the mice who died, the fog, the birds, et cetera, et cetera, et cetera. Here's just photos of some of those beautiful things that have been inspiring me over my life. Thank you to all of those. And that is the end.
Do we have, okay, that was an awesome talk, super amazing, I love the Beatles. I did have a question about the substrate and the leaf litter on top. And I was wondering, it's kind of a 2-parter question, but the first part is when you were talking about burying on leaf litter, was that, when you checked, was it burying below the leaves or below the soil, below the leaves? And if so, do you think there's a dynamic between maybe not buffering, but it just being an easier substrate for the beetles to get it under?
Yes, so that's the first question. Should I answer?
Or
that
that's
first?
all of it?
Oh, that's all of it. Okay, so yes, so I only considered burial when it was buried below the soil. And so that's when it's actually going to be protected from microbes and other scavengers, etc. And I did not bury the carcasses when they were placed in the leaf litter. It was just available. The carcass was placed on top, but kind of as an initial process, as the beetles are scurrying around the leaf litter, it's kind of getting submerged within that. But I only counted burial as when it's truly submerged into the soil. So they had the same kind of availability of soil to bury in, but then some just also had this buffering effect when it hasn't been buried yet.
Okay, thank you.
I have a bunch of questions actually. This is a super fascinating talk. I'll just limit it to one. At the very beginning, in your competition experiment, you showed that the dry carcass would only ever was buried when there was competition. Do you feel like that was just sort of a fluke, or do you think that there is something actually about the competition that is inspiring them to be like, oh, this is a good one, or I better get this underground because other things are,
yeah, no, definitely. I do think that, because they're kind of enclosed within these chambers, so the competition is almost stronger because of that. They can't escape. The nitrophores were like they were perceiving that, and so they were more like, oh, we just have to get this below ground, protect it. The heterostiltha do not like, they don't bury or really get super below. And they can like process, you know, figure out if it's actually worth. This is all just me anthropomorphizing them, but yeah, I do think that that could have been a factor there. Oh, sure, you have a question.
This was so cool. It was so amazing. I have a question about, so the fog provides moisture to the carcasses, but does like rain have a similar effect, or like, my question is about
Yeah.
rain. What's up with that?
Yeah, rain, yes. Rain will like mess up the fog collectors because, when they get too much. But so their breeding period is pretty much like June through like maybe into September. So over that time, there's usually not a ton of rain, at least this environment. So so that moisture from fog is like the only moisture they're really able to access on the coast, you know. Else it would be like inland where it's really dry and hot and everything. So but like in other environments where rain is common during their breeding season, that could totally be an effect, whether detrimental or positive.
Super cool talk. I have a follow-up based on what you just said.
Yeah.
Is this, when you say in other environments, is this like I imagine Nicropus is like wider spread, but like particular species, like is there adaptation to this environment happening? Like, do you think roughly the same beetle would encounter like totally different environmental regimes, or do you think they're like kind of tailored to this foggy?
Yeah, so I do think that it seems that with my experience, there is some local adaptation for this species to an environment like at Bodega, but even the range of that focal species is across a lot of the Western US. So in other places, I will say, like, based on just looking at my naturalist like observations and stuff, they do usually seem to be somewhat confined to like maybe like higher elevations or like along riverbeds. So still having some kind of moisture limit there. But I definitely think there's like individuals that have different capacities based on their environment in terms of moisture.
Could you talk more about maybe what you think some of the mechanisms are, why the moisture is so important, or how the desiccation of the carcasses, why that has such a big effect? And I feel like it has to tie in somehow with like how the environmental aspect as well, like populations of the It's really, really interesting.
Yeah, so I think like in my third chapter, I kind of started thinking more about mechanism because, you know, I was just depositing a fog, you know, addition like at the first few days, like nothing has happened yet in terms of like burial and everything. So I do think moisture is really important to them below ground. And so part of sequestering that carcass away and getting it below ground is to kind of preserve that, or else it's like dehydrating quickly, you know, in the elements. And moisture is, I think, super important for like the development of the larvae, especially just like going even from like larval to pupation. If there wasn't enough moisture in the soil, that just didn't go well. So I think even also like, just as the larvae is growing and shedding and everything, like there's more of that potential there. But it's kind of interesting that there is like such a big effect. And so, mechanisms.
That did actually lead to one of my last questions, which is you had a hypothesis that there would be more beetles later in one of them. It was like 19 and 27 days, and they were actually larvae. There are way less larvae. Do you think that's basically because when it's not as moist, the larvae are dying?
Yeah.
And it's like attrition that causes that difference?
Right. Yeah. So I think that's possible. And so like for that, so these were like separate cohorts, you know, I wasn't checking them as I went. I wish I could have checked them as I went, but when you check them, it kind of destroys everything. But I do think that it's likely that there were some successful like more successful larvae in those. And then just with time, since enough moisture wasn't sequestered, like they couldn't continue feeding, like there maybe was just less resource generally, like just less nutrients available because of that lower moisture. Thanks for staying.